Good morning, good afternoon, and good evening to our friends all over the world. First, I would like to thank the meeting organizers, especially Mr. Bernie Siegel, for inviting me to the summit. I would like to congratulate Mr. Siegel for all your dedication and all your wonderful work for the community of stem cell and regenerative medicine throughout the years, and I'm very proud to be part of this conference. Today, I would like to share with you some of our work at the Shanghai Institute of Medical Genetics for the past 4 decades for the advances and progresses for diagnosis and treatment of beta-thalassemia. First of all, hemoglobinopathies are the most common autosomal recessive disorder occurs worldwide. 7% of the global population carry an abnormal hemoglobin gene. More than 500,000 affected children are born each year. Hemoglobinopathies includes all genetic hemoglobin disorders. Hemoglobin is a tetramer with 2 alpha and 2 beta chains. Mutations and/or deletion in the alpha or beta globin genes will lead to hemoglobinopathies, including thalassemia syndromes and structural hemoglobin variants, known as the abnormal hemoglobins. When gene defects cause the hemoglobin synthesis disorders, this gives rise to thalassemia. When they cause changes in hemoglobin structure, this gives rise to abnormal hemoglobin. In the early 1980s, a large-scale survey organized by the Chinese Society of Genetics involving 900,000 people across 28 provinces in China was conducted to determine the prevalence of hemoglobinopathies and thalassemia in China. This survey filled the gap in China's abnormal hemoglobin analysis in the world at the time. Results of this survey showed a high population frequency of thalassemia in the southern region of China. Particularly, the incidence for alpha thalassemia was highest in the Guangxi Zhuang Autonomous Region and Guangdong Province, which also has a high incidence of beta thalassemia. The chemical structure of 59 hemoglobin variants were identified in about 700 families in China. Among them, 20 are new variants, including alpha, beta, and gamma mutations. They were named after cities they were discovered. Today, I would like to focus on beta-thalassemia. Beta-thalassemia is a recessive monogenic hematological disease caused by deficient synthesis of the beta chain of hemoglobin. The imbalances in the alpha-beta ratio of the hemoglobin heterodimer can produce the various symptoms associated with the disease, such as anemia, splenomegaly, extramedullary hematopoiesis, ferric iron deposits, and tissue damage. The highly variable clinical manifestations range from mild hypochromic anemia to moderate hematological disease to severe lifelong transfusion-dependent anemia with multi-organ involvement. The earliest report of a molecular diagnosis of thalassemia came from Dr. Y.W. Khan in 1976 using liquid DNA-DNA hybridization. Our institute reported then DNA dot blot hybridization in 1985 for the prenatal diagnosis of thalassemia, and later southern blot analysis, PCR-based ASO, and MAS techniques. Today, we can use real-time PCR and next-generation sequencing and so on. The diagnosis techniques have evolved over the years. Our institute was founded in 1978 by Professor Fanyi Zeng and Professor Shuzhen Huang about the same time that we started the studies on hemoglobinopathies. Over the past 4 decades, we have been continuing our research in the diagnosis and treatment of beta-thalassemia. Here are some representative work which I will show you, especially our efforts in exploration of treatment of beta-thalassemia. Traditionally, severe beta-thalassemia patients mostly rely on lifelong blood transfusions combined with iron chelation. Some will later develop cardiac failure and leading to death. hematopoiesis stem cell or cord blood transplantation are also used as a strategy for partially relieving the symptoms. However, resources for finding matched bone marrow donors are limited. All of the aforementioned therapies are expensive and can be painful procedures. With the development of stem cell induction and gene editing techniques, gene or cellular therapy and autologous stem cell transplantation potentially represent an excellent approach to treat genetic diseases. Our early attempt were using drug therapy with hydroxyurea proved to alleviate the alpha-beta imbalance in the beta-thalassemia intermediate patients. With the development and maturation of new technologies, we started the exploration of treatment of beta-thalassemia through cell and gene therapy. I would like to briefly touch upon some of these approaches we investigated. We used one of the most common beta-thalassemia syndrome, beta-654 in China, that we studied extensively since 1994 to study various treatment options extensively. Beta-654 thalassemia is the most common form, which represents approximately 1/5 of total beta-thalassemia cases in China. These patients are presented with severe anemia, with abnormal hematologic indices, including lowered red blood cells, hemoglobin, and mean corpus hemoglobin concentration, MCHC. Splenomegaly and hepatomegaly related to significant extramedullary hematopoiesis, EMH, as well as iron deposits in multi-organs are typically observed. We identified the molecular defects for beta-654 thalassemia. That is a C2T substitution at nucleotide 654 of intron 2 in the beta-globin gene, which could lead to an incorrectly spliced RNA isoform that produces trace amount of a poor functioning polypeptide. Fortunately, a human beta-654 mouse model was generated while the 1 murine adult beta-globin gene was replaced by a single copy of the human beta-654 gene. The mouse displayed similar symptoms as the beta-654 thalassemia patients: severe anemia with abnormal hematological indices, splenomegaly, and hepatomegaly related to significant extramedullary hematopoiesis, EMH, as well as iron deposits in multi-organs. This serves as an ideal animal model for studying the therapeutic effect of gene and cell therapy of beta-654 thalassemia. Using RNAi to interfere with alpha-globin gene expression, while simultaneously blocking the beta-654 aberrant splicing site in the beta-64 thalassemia mouse model, we achieved an improved alpha-beta globin chain balance, leading to a decrease in the excessive participation of alpha-globin chains around the cell membrane and in turn a recovered phenotype. We also first reported the lentiviral vector delivered of a beta-globin gene to treat beta-thalassemia in mouse model and observed phenotypic recovery in the beta-654 thalassemia mouse. From gene therapy, we moved to combining gene and cell therapy. Induced pluripotent stem cells, iPSCs, has recently been considered an excellent approach to treat many diseases, including genetic diseases. As such, we reported in our effort using chimeric murine models to evaluate the in vivo efficacy, safety, and stability of treatment of beta-thalassemia through iPSCs that were derived from the beta-654 mice and corrected with gene modification using normal human beta-globin gene. We first produced iPS cells from beta-654 thalassemia mice and verified their pluripotency using in vitro and in vivo analysis. iPS cells were generated from normal mice with the reporter genes. Various chimera mice were produced through blastocyst injection into beta-654 mice using normal or beta-654 iPS cells, corrected with gene modification of normal human beta-globin gene. These blastocysts were transferred into pseudo-pregnant wild-type mice, which gave birth to chimeric pups at approximately 20 dpc. Analysis of the beta-654 chimera mice demonstrated that all beta iPSC chimeras had stable transgene expression of the human beta-globin mRNA. In addition, the iPSCs used could differentiate into multiple tissue types with varying efficiency. Also, the hematological parameter of treated chimeric mice exhibited elevated levels of RBC, hemoglobin, MCV, and MCHC compared to the beta-654 mice, showing a promising therapeutic effect. Further examinations display reduced clinical symptoms of severe anemia with abnormal hematological indices, splenomegaly, significant extramedullary hematopoiesis, as well as iron deposits. Interestingly, a dose effect was observed. When exogenous beta-globin were expressed at an over 30% level, there was a significant therapeutic effect on beta-654 thalassemia mice. But if the beta-globin were at the less than 20% level, certain degree symptoms of thalassemia remained. This could provide indication on the dosage effect for the gene or cell therapy in beta-654 thalassemia patients. One of the important tasks was to test the efficiency and safety of the gene and cell therapy approach used. This is an example of the strategies when we first test in the mouse model before it could be used in human patients. Over the past decades, multiple in situ gene editing techniques have been developed that could be applied to improve the effectiveness of gene therapy. We have utilized TALENs and CRISPR-Cas9 approaches. Our studies using these gene modification methods to correct genetic defects of the beta-thalassemia mice both showed promising effects. We also explored one other exciting treatment option for prenatal treatment for beta-thalassemia patients, the in utero transplantation using allogenic or zygogenic hematopoietic stem cells. We first developed the in utero transplantation to study the stem cell biology in vivo under normal physiological conditions in goats. As the fetuses were incapable of rejecting transplanted allogenic cells because of its immunological incompetency, the non-self stem cells could be tolerated by their immune system. The human hematopoietic stem cells were able to survive and differentiated into various tissues and organs of in utero transplanted goats, and a substantial fraction of human cells engrafted in goat livers and other tissues and expressed human proteins. We can now employ the in utero transplantation of stem cell to test the therapeutic effect in beta-654 mouse model and to observe the stability of long-term treatment effects. GFP-labeled stem cells were injected into 13-day-old fetuses of beta-654 thalassemia mice, and multiple analyses were performed. Fluorescent microscopy displayed donor GFP-positive mice cells are chimerized in intrauterine transplanted mice. Pathological analysis also displayed fewer numbers of erythroid precursors in the spleen red pulp, and the marginal zones were distinct. And the hemosiderin in the livers of the transplanted beta-654 mice was markedly reduced than that in the beta-654 mice. Extramedullary hematopoiesis was rarely found in the transplanted beta-654 mice. Liver pathology and iron accumulation was dramatically reduced in transplanted beta-654 mice. Splenomegaly was significantly reduced in recipient beta-654 mice also. Beta-654 mice after in utero transplantation exhibited markedly improvements in hematological indices such as RBC, hemoglobin, MCV, MCHC. Peripheral blood RBC counts and hemoglobin levels were very much improved. Furthermore, recipient mice still showed continuous therapeutic effect 8 weeks after. The cell and gene therapy efforts for the treatment of thalassemia is continuing, and there is still much to be done. Ultimately, the safety, effectiveness, and affordability of cell therapy and gene therapy must be improved in order to provide patients suffering from beta-thalassemia with the best therapeutic options. Today, I share with you our efforts in the diagnosis and treatment of beta-thalassemia at Shanghai Institute of Medical Genetics for the past 40 years in a nutshell. And we will continue to explore better and more effective means to help our patients and contribute to the scientific community. I would like to thank the founders of our institute, Professor Yi-Tao Zeng and Professor Shuzhen Huang, and all the wonderful people that contributed to this work in our institute. Finally, we extend our sincere welcome for you to visit us in Shanghai in the near future. Thank you.